Hello everyone, and welcome to today's live webcast, Next Generation Sequencing for BRCA1 and BRCA2 Mutation Testing. My name is Soon Pham, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the live Q&A event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem to the green Q&A button lower left. And now a brief note about QIAGEN, our sponsor for today's web event. QIAGEN is the leading global provider of sample-to-insight solutions to transform biological materials into valuable molecular insights. QIAGEN sample technologies isolate and process DNA, RNA, and proteins from blood, tissue, and other materials. Assay technologies make these biomolecules visible and ready for analysis. Bioinformatics software and knowledge bases interpret data to report relevant, actionable insights. Automation solutions tie these together in seamless and cost-effective molecular testing workflows. QIAGEN provides these workflows to more than 500,000 customers around the world in molecular diagnostics, applied testing, pharma, and academia. And now I would like to introduce today's speaker, Dr. Raed Samara. Dr. Raed Samara is the Global Product Manager responsible for next-generation sequencing at QIAGEN. Before this, he was scientist in the R&D project management department of QIAGEN at Frederick, where he manages and coordinates multiple life sciences product development projects across R&D and operation teams. Prior to joining QIAGEN, he was a postdoctoral fellow at the National Cancer Institute, conducting research in the field of cancer immunology, with emphasis on identifying strategies to boost efficacy of cancer vaccines. He received his PhD degree from Georgetown University in tumor biology. I will now turn over to Dr. Raed Samara for his presentation.
Unmuted.
Thank you, Soon. And just a couple of legal disclaimers before I get into this webinar. The QIAGEN products shown in this webinar are intended for molecular biology applications only. These products are not intended for the diagnosis, prevention, or treatment of a disease. And for up-to-date licensing information and product-specific disclaimers, please see the respective QIAGEN kit handbook or user manual. All of these handbooks or user manuals can be found at www.qiagen.com. Okay, so as an outline for today's webinar, first I will go through an introduction on BRCA1 and BRCA2 genes, just to give you a general background. And then I will introduce to you the sample-to-insight solution that QIAGEN has built for next-generation sequencing challenges. Then I will go through some performance and applications data. I will give you two examples, one for the application of routine testing, and then the second one is for the application of biomarker studies for disease stratification in clinical trials. And then I will close this off with a summary. So on to the first topic, introduction to BRCA1 and BRCA2 genes. So it is known that BRCA1 and BRCA2 genes are key tumor suppressor genes. They have DNA repair and transcriptional regulation functions. Their importance is highlighted in breast cancer. We all know that mutations in BRCA1 and BRCA2 genes can predispose both females and males to the development of breast cancer. As a matter of fact, around 0.25% of U.S. women carry a mutation in BRCA1 in some BRCA genes. This translates to around 375,000 cases a year. And these mutations will put these individuals at a very high risk for hereditary breast and ovarian cancer. BRCA mutations are associated with 40 to 80% lifetime breast cancer risk, and they are associated with about 11 to 40% lifetime of ovarian cancer risk. Now, the importance of BRCA1 and BRCA2 mutational testing is highlighted by the fact that knowledge of the risks of breast cancer and ovarian cancer allows us to prevent those diseases, and also knowledge of the mutational status of BRCA1 and BRCA2 can also help us in optimized disease management. And I will give you again an example of how those studies can be used in clinical trials. So for BRCA1 and BRCA2, there has been new recent developments. The BRCA1, BRCA2 mutational status has been studied in breast cancer, and the effects of a PARP inhibitor drug known as olaparib, has been shown to as maintenance therapy to be associated with increased progression-free survival versus placebo in patients with platinum-sensitive recurrent ovarian cancer. So those patients who have shown mutations in their BRCA1 or BRCA2 genes respond more favorably to the PARP inhibitor than those with wild-type BRCA1 and BRCA2. And this is clearly shown in this paper that was published late last year. So when the authors of this study looked at the response of all patients versus BRCA mutated patients versus BRCA unmutated patients to this PARP inhibitor, otherwise known as olaparib, they did see that there is an increase in the response rate in those patients in which BRCA1, BRCA2 are mutated compared to those patients in which BRCA1 and BRCA2 are unmutated, indicating that mutations specifically in BRCA1 and BRCA2, which belong to these the tumor suppressor genes and involved in DNA repair machinery, mutations in these genes will allow those patients to respond more favorably to inhibitors of PARP. And so this was really a key study that highlighted the importance of looking at the mutations of BRCA1 and BRCA2 in ovarian cancer specifically. But as I will show you later on in this webinar, you can actually see that mutations in BRCA1 and BRCA2, as well as other key tumor suppressor genes and genes that are involved in DNA repair, are also important for the response of the same against the same drug in prostate cancer patients as well. And because of these results, this olaparib drug received a positive opinion in the EU because of its efficacy in ovarian cancer. So onto the sample-to-insight solution that QIAGEN has built for next-generation sequencing challenges. So I mentioned that the mutational status of BRCA1 and BRCA2 are important to know from both a therapeutic perspective as well as from an understanding the underlying mechanisms of a disease perspective. And so, but we know that to use next-generation sequencing for studying the mutational status of BRCA1, BRCA2, and other genes as well is very challenging, because there are a lot of challenges associated with the next generation sequencing workflow, from sample preparation to data analysis. And what I will go through next is the sample to insight solution that QIAGEN has built to address these challenges. So QIAGEN has built this universal workflow from sample to insight to overcome NGS challenges. I've broken down the NGS workflow into 6 individual modules, if I may call them, and 3 individual QC modules as well. And what I've done here is listed the challenges associated with each one of these modules, just to kind of give you an example of what it takes and what kinds of challenges you will go through along the way. So it all starts with sample isolation. Sample isolation can be followed by targeted enrichment if you are interested in undertaking a targeted DNA sequencing approach. This is where you enrich your sample for specific genes or specific regions of interest, as opposed to a whole genome or a whole exome approach, where you're either looking at every single base in the genome, or you're looking at specifically the exon coding regions of the genome. This is followed by the library construction step. That's where we construct libraries from those regions that we have enriched in the targeted enrichment step, followed by the NGS run, and then data analysis and interpretation. This is also a major challenge. There's a lot of data that gets generated through from NGS runs. These data have to be analyzed and also interpreted. The interpretation step means just what, if you find a mutation in a specific gene, what does it mean from a biological and clinical perspective? So again, this is just a breakdown, a general overview of the workflow associated with almost any NGS run for targeted enrichment. Now, when it comes to challenges, there are a lot of challenges, as you can see here. For sample isolation, there's the challenge of isolating high-quality DNA samples. So we know that DNA can come from many different types of samples. A lot of researchers use FFPE samples, and we know that the DNA quality of these samples is extremely low. You can end up with very highly fragmented DNA, and you can end up with very low amounts of DNA. So this brings me to the second challenge for sample isolation, which is quantifying amplifiable, not bulk amounts of DNA. Because PCR is used in almost every step of this workflow, you want to make sure that the DNA that you're using is amenable to amplification by PCR. A lot of the methods, as you will see throughout this webinar, a lot of the methods just quantify bulk amounts of DNA. The main challenge here is that if you only quantify the bulk amounts of DNA, you might be actually overestimating the amplifiable amounts of DNA that you have in your sample, because some of that DNA might not actually be amplifiable through PCR in this workflow. For the targeted enrichment step, the main challenges are turnaround time and limited amounts of DNA. You don't want a targeted enrichment step that takes days to finish, and you also want to be able to enrich your targets using very limited amounts of DNA. Again, going back to the FFPE example, if you have a couple of slices of DNA, a couple of slices of the sample, how much DNA can you actually isolate from these samples? It's going to be very limited. So you want a targeted enrichment approach that allows you to use these limited amounts of DNA. When it comes to library construction, the main challenge is library yield and the multiple steps that are needed for library construction. And I will show you how this sample to insight solution has addressed these challenges. For the NGS run, each platform has its own specific challenges. I'm not going to go through these challenges in this presentation or webinar. There's a lot of material on the web that you can check out if you're interested in one platform versus the other. For data analysis, again, there's a lot of information and a lot of data that gets generated from next generation sequencing. Analyzing this data is a major challenge because all the reads that are coming off of a sequencer have to be appropriately mapped and aligned, and then variants have to be called off of these reads. The interpretation part is also challenging because you have a lot of mutations that get detected in these samples, and the challenge here is to identify those that are actionable versus not actionable. Which ones can you act upon? Which ones can you not act upon? Or you do not have enough information to act upon. So again, this presents another challenge. So what I will go through today is to show you how this sample to insight solution addresses some of these challenges along the NGS workflow. And also along that workflow, I will be talking about specific products that QIAGEN has developed that will fit into the individual modules. And again, remember, all of these kits that I will be mentioning today are for molecular biology applications only. So starting off with challenging samples or DNA from challenging samples. So just to kind of give you an example of FFPE samples, we know that the use of DNA isolated from FFPE tissues remains a major challenge. Why? Because the quality of DNA is very low from FFPE samples. There's a lot of base damage because of the preservation process. And this ends up with an increased rate of false positives. There are many papers out there that have described these artifacts and these challenges. This is one of them. It was published in BMC Medical Genomics. If you're interested in accessing it, it is freely available on the web, and it talks about the challenges of using FFPE samples. So what QIAGEN has done is it has developed the GeneRead DNA FFPE kit. The GeneRead DNA FFPE kit enzymatically removes artifacts during DNA isolation steps and provides more confidence in SNP calling or variant calling. So it actually, it physically and enzymatically removes these artifacts that you can see in FFPE samples. What this translates to is improved accuracy in variant calling. And I will show you an example on the next step. Another feature of this kit is that it can be automated on the QIAcube to streamline your process. So here's an example of how this kit can be used and the value that it carries. So what you're looking at here is some variants that have been detected in 15 FFPE samples. These are actually not 15. This is one sample, but it's 15 years old. It is a 15-year-old liver carcinoma FFPE sample. So the sample was isolated, or the DNA from this sample was isolated by two different methods, either the QIAAMP FFPE kit or the GeneRead FFPE kit that enzymatically removes these false positives. Once the DNA was isolated, what we did is we enriched 160 genes that are heavily implicated in cancer using the GeneRead Comprehensive Cancer Panel. We constructed libraries to be sequenced on an Illumina platform, specifically the MySeq. So what you're looking at here are the different variants that have been detected in some of these genes in this 15-year-old liver cancer sample, specifically FFPE sample. And what you see in the red and green cells are those mutations that have been corrected for by the GeneRead FFPE kit. We know that these are false positives that have been corrected for by the enzymatic removal of these artifacts in the GeneRead FFPE kit. So if you look at this C2T mutation that was seen using QIAAMP FFPE kit, you do not see that mutation now in the GeneRead FFPE kit. And this is true for some of the mutations, for a C2T mutation or a G2A mutation as well. However, we know that not all of the G2A or C2T mutations are false negatives. I mean, some of them are true positives, and you actually see those types of mutations not being corrected for because they are true positives. However, for those ones that are not true positives, you see them being corrected for. So this is just an example of how the enzymatic removal of these artifacts can actually increase your accuracy of variant calling in your FFPE samples. Now, another challenge, as I mentioned, is the quantification of DNA. So not all DNA that you will isolate from your samples is amplifiable and is functional in the NGS workflow. So the inaccurate quantification of input DNA is a major challenge because it can lead to poor target enrichment and NGS library yield, eventually leading to wastage of your precious samples and time, and also wastage of your money because you've just spent your sequencing run on DNA that cannot be sequenced. So to highlight this challenge, I just kind of want to touch upon a couple of ways through which DNA is usually quantified. The two major and most commonly used methods is spectrophotometry and fluorometry. Spectrophotometry measures any form of DNA, while fluorometry can measure double-stranded DNA down to 20 bases. Now, while these are very helpful for specific samples, the quantification by these methods can be a major challenge, simply because what you are measuring is bulk amounts of DNA. You're not measuring the amplifiable amounts of DNA that are present in your samples. You're just basically taking a look at every single form of DNA that exists in these samples. Again, this is a challenge because the non-amplifiable DNA is not going to be able to participate in the different steps of the NGS workflow. One way around this is to use qPCR. We know that qPCR is very accurate, and it gives you a good representation of the amount of amplifiable DNA that is present in your sample. We have taken advantage of qPCR and developed the GeneRead DNA Quantum Eyes kit to specifically address this challenge. It is a qPCR-based kit that measures amplifiable or functional DNA. By using this kit, you can actually get a good sense of how much of that DNA that is present in your sample can be amplified by PCR and can be translated to library yields and good sequencing data. The GeneRead DNA Quantum Eyes kit uses 2 qPCR assays to query more than 40 genomic loci that are randomly distributed in the genome. So because we are targeting more than 40 genomic loci that are randomly distributed, it gives us a good sense of the amount of fragmentation that is present in those DNA samples. And not only does it give us a good quantification and qualification of the PCR that is amenable to amplification, the DNA that is amenable to amplification, it also gives us guidance to rescue those low-quality DNA samples. Specifically, it's going to give you the appropriate amount of input DNA that is needed for that specific sample if you want to translate that DNA into appropriate library fragments and NGS sequencing data. It also gives you the appropriate number of targeted enrichment cycles that must be used in your targeted enrichment step if you want to be able to use that specific sample, that specific FFPE sample, call it, for sequencing. And what this translates to is it ensures reproducible high post-amplification yields. Because it is a qPCR assay, it's a very simple setup whereby the genomic DNA, once it's isolated from FFPE samples or any of those low quality yielding samples, the genomic DNA is mixed with the qPCR assays and master mix. You run qPCR, and through a simple data analysis setup that is available through QIAGEN, you actually get the amounts of DNA that are amplifiable in your sample, and also get this appropriate number of targeted enrichment cycles that are needed for that specific sample. To give you an example of how this kit can be used, what we've done here is we've run 15 different samples. We've quantified the DNA from these samples by 2 different methods, spectrophotometry and qPCR using Quantum Eyes. And what we looked at next is the library yield on the left and the sequence specificity shown on the right. So when we look at the library yield of those samples, if they were quantified by spectrophotometry methods, you can see in the blue diamonds that the library yield is pretty much all over the place. You get some samples that have not given any yielded any library fragments, whereas some that have yielded some library fragments, but not optimal amounts of library fragments. But if you look at the same samples, if they were quantified by Quantum Eyes, as shown in these purple circles, you do see a pretty consistent library yield from the different samples. Remember, these are 15 different FFPE samples, and because they were quantified by Quantum Eyes using a qPCR approach, we were able to accurately quantify the DNA, resulting in consistent library yields from these 15 different FFPE samples. When we look at the sequence specificity, so these are the specificity of reads that are coming out of the NGS run on a MiSeq. We can also see some major differences between those DNA samples that were quantified by spectrophotometry versus those that were quantified by Quantum Eyes. You can see a very high degree of specificity for those reads that are coming off the samples that were quantified by Quantum Eyes, as you can see in the blue circles, versus the blue diamonds where those DNA fragments were quantified by spectrophotometry. So again, it's a very helpful kit in order to quantify the amplifiable amounts of DNA. So now that you've quantified your DNA, the next step would be to enrich for specific genes. This webinar is focused on BRCA1, BRCA2, but the same principles can be applied for other genes in the human genome. What we have done is we've taken advantage of the multiplex PCR based system to allow us to design primers against the specific genes that you want to enrich. Again, because this is a BRCA1, BRCA2 webinar, I'm going to use the example of BRCA1, BRCA2 genes. So with targeted enrichment, the major challenge is that you want to sequence every base within your targeted region with little amounts of DNA. And that led us to the development of the GeneRead DNAseq Targeted Panels version 2. They use a multiplex PCR based enrichment. So instead of using a hybridization-based approach followed by PCR, we go straight to PCR to enrich for the genes. It provides, so with the kits or with the panels, we provide primer sets for any region, any gene, or any set of genes in the human genome. So you can define the region the way you need to according to your requirements. That could be entire genes. It could be just certain exonic regions of genes. It could be specific introns or promoter regions, and so on and so forth. The panels will provide overlapping primers, and those primers are divided into an appropriate number of PCR pools. In the case of the BRCA1 BRCA2 panel that I will focus on today, they have been divided into 4 PCR pools. So when I talk about a panel, it's basically a collection of primers in PCR pools. So why did we choose the multiplexed PCR-based targeted enrichment approach? There are 2 major approaches for targeted enrichment. You have the multiplex PCR-based approach, and then you have the hybridization-based approaches. Each one has its advantages and disadvantages, but the main reason why we decided to choose the multiplex PCR-based approach is because of the specificity and uniformity of PCR that cannot be matched by other targeted enrichment approaches. This was nicely highlighted in a Nature Methods paper that was published back in 2010 that actually compared to the different targeted enrichment approaches from multiplex PCR to hybrid capture or MIPS. If you're interested, I highly recommend going through this paper. It has done an excellent job of comparing the different methods. But really, one of the conclusions that came out of this paper is that the specificity and uniformity of PCR for targeted enrichment cannot be matched by any other approach, and that is the reason why we decided to go with a multiplex PCR-based targeted enrichment approach for our panels. So we've developed a 15 different cataloged panels. Again, just to remind you, the panels are basically a collection of primers that target specific genes or regions of interest. So the 15 different panels that we have developed have different applications. So we've developed some for solid tumors, like the clinically relevant tumor and the tumor actionable mutations panel. We've developed a panel for hematologic malignancies, which is the myeloid neoplasms panel. We've developed 9 other tissue-specific panels for breast cancer, colorectal cancer, liver cancer, and so on and so forth, as you can see on this slide. And we've also developed comprehensive panels in the form of a cancer predisposition panel, the comprehensive cancer panel, and the carrier testing panel. You also have the option of building your own custom panel. So we do have 2 different ways by which we can construct your custom panel, depending on what types of targets you're interested in targeting. But today's focus is on BRCA1 and BRCA2, and QIAGEN has also developed a BRCA1, BRCA2 targeted panel that specifically enriches for the exonic regions of BRCA1 and BRCA2. Now, where did the content of these panels come from? It came from different databases. We've tapped into multiple databases to build the content for these panels, from the Cancer Genome Atlas to COSMIC to even private databases such as the Ingenuity systems. So a lot of databases have been tapped in order to build the content of these panels, and all of these panels, except for the custom panels, have been Workbench tested in the lab in-house here. So, zooming onto the BRCA1, BRCA2 panel. So, as I mentioned, that this panel is specific to the exonic regions of BRCA1, BRCA2. It has a lot of neat features and benefits. The amount of input DNA needed is very minimal. You only need 40 nanograms of DNA. So, even if you're working with FFPE samples or these other types of samples that yield very low amounts of DNA, you can still enrich for your genes using this panel. It utilizes an overlapping amplicon design, so all the amplicons that we design are overlapping to maximize the coverage for all the different exonic regions in BRCA1, BRCA2. In 8 hours, you can construct your libraries, and the other feature of this panel is that it yields 1 library per sample. You're not constructing libraries from each individual PCR pool. What you end up doing is you run your PCR in 4 pools, but then you end up pooling all these amplicons together, going into the library preparation step. So although you're enriching using 4 pools, what you end up with is just 1 library per sample, which cuts down on your costs. Now, looking at some specifications, it has a 100% coverage for every single base in the exonic regions of BRCA1 and BRCA2. Not only do we target the exonic regions, but we also target some intronic regions to cover the intron-exon junction. So we cover, on average, about 20 bases of around each exon. So again, we cover the exonic regions plus some of the intronic regions to get that intron-exon junction. There are 21,000, close to 21,500 bases covered by this panel. The average amplicon length is 150 bases. So the amplicon length is also an extremely important feature. The amplicons are small enough to make them compatible with FFPE samples. The number of primer pools is 4. So again, as I mentioned, you will enrich your DNA samples in 4 PCR setups, but then what you end up doing is pooling all these primers or amplicons to generate only 1 library per sample. 40 nanograms of DNA is needed. The number of amplicons needed to cover all the exonic regions of BRCA1 and BRCA2 is 250. So it's still a fairly small panel that gives you the power of high multiplexing. So if you want to run many more samples on an NGS run, you can easily do so because the targeted region and the number of amplicons is small. It has a high degree of specificity and uniformity. So we're seeing 99% specificity, which means that 99% of the amplicons that are generated are specific to the targeted regions that we wanted to target, which is basically BRCA1 and BRCA2. Uniformity is very high, so it allows you to make better use of your sequencing capacity. So you're not really wasting a whole lot of sequencing capacity on unwanted or ununiform regions. And the percent of bases called is 100%. So we're covering every single base in the exonic regions, as well as those 20 base pairs around the exonic regions of BRCA1, BRCA2. And this is, since we are talking about a tumor suppressor gene or tumor suppressor genes, it is important to cover every single base because as opposed to oncogenes which have mutations in certain hotspots, tumor suppressor genes tend to accumulate mutations across the entire length of the gene or the coding regions of the gene. So when you're looking at tumor suppressor genes, it is extremely important that you cover every single base of those genes. So to give you an example of the overlapping nature of the amplicons, what you're looking at here is just a snapshot of from IGV, where I've included the design of amplicons, and I've included some performance data on the PGM as well as the MiSeq. So what you're looking at here, you're looking at the coverage of this specific exon of BRCA1. In the NA12878 DNA sample. So this is the genome in a bottle sample that is being developed as a standard for NGS runs. So we've enriched BRCA1 and BRCA2 from this DNA, and we've sequenced the libraries on a PGM. We've also processed DNA from FFPE sample, and we also processed the DNA from the NA12878 sample and constructed libraries for sequencing on a MiSeq. So again, what you're looking at here is just an example of how the amplicons are overlapping for the specific BRCA1 exon. And the same is true for all the other exons in BRCA1 and BRCA2. And I'll show you in the next couple of slides how the performance of this panel looks like on a PGM and a MiSeq. So for BRCA1, what I'm showing you here is how the different exons of BRCA1 are covered by this panel on 2 different samples and 2 different sequencers. Again, the NA12878 on a PGM, an FFPE sample on a PGM, and the NA12878 DNA on a MiSeq. We can see coverage of every single base across the different exons of BRCA1, and the same is true for BRCA2, as I will show you on the next slide. And not only that, but we do see a very high degree of uniformity uniformity across the different exons of BRCA1 for this slide here, which allows you to sequence to sequence your samples to a specific depth of coverage, and you don't have to go very high to account for these lowly covered regions. A high degree of uniformity, as you can see over here, allows you to be more flexible with your DNA, with your sequencing coverage. So here's coverage for BRCA2. Again, what you're looking at is 100% coverage of all the exonic regions of BRCA2 with a very high degree of uniformity also. And actually, you can see on this slide over here how there are many amplicons tiling across each other to cover a specific exon. Again, that goes back to the overlapping nature of the amplicons to maximize the coverage across the regions that we want to cover, and it also shows you the high degree of uniformity across different samples. So again, we've used this NA12878 DNA, which is being developed as a reference, and it's considered high quality DNA. So with high quality DNA, we can get an excellent degree of uniformity, and also when we look at FFPE samples, we still get a high degree of uniformity even with those low quality DNA. So whether you're using high quality DNA or low quality DNA, you can still get pretty good enrichment for the BRCA1, BRCA2 genes using the BRCA1, BRCA2 panel. Okay, so I mentioned the importance of covering every base in BRCA1 and BRCA2 because they are tumor suppressor genes. Now, anyone who has done sequencing with amplicons understands the challenges of sequencing the ends of amplicons. The ends of amplicons in next generation sequencing tend to be problematic regions because some of the bases towards the ends of amplicons do not get sequenced. It's just the nature of how sequencing works for amplicons. So it is important to protect the ends of those amplicons. And in order to accurately call variants near the ends of those amplicons or reads, primer sequences have to be present for read mapping and alignment. So we use a multiplex PCR-based approach that utilizes primers that will span or cover the region that we're interested in, and we'll use those primers to basically amplify that region that we're interested in. Now, those primer sequences, they are not native to the sample, so you cannot use them to call variants within those regions, but you want to keep them, you want to keep those primer sequences intact while you're mapping and aligning your reads, because basically those primers are now going to protect the ends of the amplicon, so you can actually get those one or two bases that are part of the amplicon but sit at the end of it. So if you have the primer sequences still intact and take that read throughout sequencing with the primer sequences, you end up protecting those the ends of the amplicons, which allows you to call variants that are present within the ends of the amplicons. Approaches that digest off the primer sequence can end up with false positives and negatives, as I will show you in the next slide in a paper that was published earlier this year. But the concept of these blind spots towards the ends of the amplicons have been described by our group here in a paper published late last year in BMC Genomics, where they actually describe the importance of keeping primer sequences up until the variant calling step. So you do not call variants within the primer sequences, but you want to keep them on the ends of the amplicons so you can protect the ends of the amplicons and allow every single base to be sequenced. Now, this was very nicely actually shown in a paper that was published earlier this year from a group in Germany who wanted to do a comparison between the different PCR or multiplex PCR-based approaches. So they used a panel from another vendor that does digest the primer sequences before the library construction step, compared to our workflow that does not digest the primer sequences, and we keep them all the way until the variant calling step, and we softly clip those primer sequences off the reads so we don't call any variants, but we carry the primer sequences throughout the entire reaction or the entire workflow. So what this group wanted to do is to just compare the two different approaches, and on a MySeq. So they would prepare libraries from the QIAGEN workflow or the GeneRead sample to insight workflow versus the workflow from AmpliSeq. And what they noticed at the when they looked at the data is that the sensitivity of the QIAGEN panel is much higher than that of AmpliSeq, simply because some of the variants that were not detected. So there were 5 variants that were not detected. 69 out of 74 were detected, and 5 out of 74 were not detected. It turns out that these five variants that were not detected, they were at the amplicon boundaries and primer binding sites, which indicates that the physical removal and digestion of the primer sequences will actually expose the ends of the amplicons, and what you end up with is false negatives or false positives towards these end of the amplicons because they were not sequenced efficiently. So, because they are key to tumor suppressor genes, what you want is to sequence. You want to sequence every single base in those 2 genes, and the GeneRead DNAseq BRCA1 BRCA2 panel gives you that 100% coverage in the coding regions of every single base in the BRCA1 and BRCA2 genes. And I will show you actually examples as I get to the applications data of how some customers have used the BRCA1 BRCA2 panel to look at to get 100% coverage. But it is a universal panel in the sense that you can use it for any sample or on any major sequencer. So as long as you can get enough DNA, as long as you can get 40 nanograms of DNA, whether it is an FFPE sample, a plasma sample, liquid biopsy, or single cell, as long as you can get 40 nanograms of DNA, you can use the GeneRead DNAseq panel to enrich for BRCA1, BRCA2, and then you can sequence the libraries on platforms from Illumina as well as platforms from IronTorrent. Now, sample preparation is only one part of this. You need powerful data analysis and interpretation tools. And to build that sample to insight solution, QIAGEN offers 2 major data analysis tools and interpretation. One is the CLC Biomedical Genomics Workbench, as well as the Ingenuity variant analysis for data interpretation. The CLC Biomedical Genomics Workbench is it's an easy-to-use and end-to-end solution in the sense that it's basically windows that you can build and put together to construct your workflow. It has a plug-in for the GeneRead DNAseq panels. So again, because it is amplicon-based sequencing, it needs a specific setup within the data analysis tool. To make things a lot easier for our customers, we've built this plug-in into the CLC Biomedical Genomics Workbench to make the analysis of amplicons much easier. So you get these, you can do mapping and alignment and even visualize the variants and call variants and even visualize them with the CLC Biomedical Genomics Workbench. Now, once those variants have been called by the CLC Bio platform, what you could do is take all that data and process it through the Ingenuity databases, through the Ingenuity variant analysis platform. This is a platform it's basically a knowledge base that is 15 years in the making. A lot of people have contributed to this knowledge base. They've looked at all the publications that exist out there and have taken all that knowledge and have dumped it into this tool. So it's really a huge database of all the variants that have been reported in the literature. So what you'll do is you'll take those variants that have been detected by and called by the CLC Bio platform and put it into IVA to start making sense of these variants. So if you see that KRAS mutation in your sample, what does it mean from a biological and clinical perspective? And not only does it tap into the Ingenuity knowledge base, but it also taps into HGMD also. So just to kind of go through some performance and application data. Again, the first example I want to show is from routine testing. So you can use this BRCA1 BRCA2 panel for routine testing of samples. But before I get into this, I kind of want to just show you or go through a brief sort of introduction to the different applications that one can do for the different panels using NGS. We know that you can look at the applications in two ways. You have detection and discovery. The clinical research area generally tends to want to do more detection, where they want to detect those mutations that are known to be actionable. When we look at translational and discovery research, there's a lot more discovery. They're interested in knowing more about their samples. Now, there's each one again has its advantages and disadvantages. If you're looking at a detection type of application, because the target size tends to be small, again, you're looking at those actionable mutations, you can do a very high degree of multiplexing. In the discovery side of applications, your target size generally tends to increase, so your multiplexing capabilities decrease. So you look at a lot more, your multiplexing capabilities decrease. If you're looking at less targets, your multiplexing capabilities increase. So again, but you could use panels to look at for clinical research as well as translational research. And these are the two examples that I will show you today. I'm going to show you how some customers have used the BRCA1, BRCA2 panel for clinical research, for mainly, again, routine testing, whereby they're interested in detecting known mutations that exist in BRCA1, BRCA2, but also how some customers have used BRCA1 and BRCA2 containing panels for translational purposes. And when I talk about a BRCA1, BRCA2 containing panel, it's basically a panel that has the same gene designs that are contained in the BRCA1, BRCA2 panel. The difference here is that you have additional targets, additional genes that the customer is interested in targeting. So to focus first on the clinical research and the routine testing aspect of BRCA1, BRCA2. So this first study is from Dr. Normano's group in Italy. Basically, the goal here was to verify the presence of BRCA1 and BRCA2 mutations in blood samples from hereditary breast cancer cases. So 6 samples were analyzed, 5 blood samples and 1 control genomic DNA. The samples were enriched for BRCA1 and BRCA2 using the GeneRead DNAseq panel, and then subjected to PGM sequencing using this QIAGEN workflow. And so looking at the sequencing metrics, we were seeing the same exact sequencing metrics that we see internally. We see a very high degree of specificity and uniformity, and every single base in BRCA1 and BRCA2 were called in these samples. And when we look at the different mutations that were detected, basically, there are certain mutations that were previously detected by Sanger sequencing. We were able to call every single one of these mutations that have been described by Sanger sequencing in these samples. However, when the same samples were run by a different multiplex PCR-based enrichment approach, specifically the AmpliSeq workflow, we know that from the customer, we know that some of these mutations were not detected in the samples where they should have been detected. So basically, because again we are covering every single base in BRCA1, BRCA2, we're able to call some of these mutations that were not called by the AmpliSeq panel. And just drawing from Dr. Normano's testimony here, the complete coverage provided by the BRCA1, BRCA2 panel allowed us to detect mutations that were missed by other targeted enrichment technologies. Again, the complete coverage of BRCA1, BRCA2 is needed because these are tumor suppressor genes, and you want to be able to call every single base in them. The second study is from a group from Dr. Butner's group in Cologne in Germany. And again, the idea here was the same, is to verify the BRCA1, BRCA2 panel performance, but the difference here is that the samples that were used in the study are FFPE samples, and the sequencing was done on a MySeq. So looking at how the different mutations could be called by the AmpliSeq panel as well as the GeneRead panel, what the customer has done here is they've run 3 runs, run 1 and 2 using the AmpliSeq workflow, and then run 4 using the GeneRead workflow. So run 4 was basically a repeat of run 1 and 2. The difference is just the workflow that was used. You have 44 samples that were interrogated, 35 ovarian cases and nine mammary or breast cancer cases. And again, the idea here was to compare the different multiplex PCR-based approaches. So when the authors looked at the number of mutations that can be detected in BRCA1 and BRCA2, they were seeing that 4 out of 21 mutations could not be called in BRCA2 using the AmpliSeq workflow. Again, simply because not every base is covered in the BRCA1 BRCA2 AmpliSeq panel. However, when you look at how the GeneRead DNAseq panel covered BRCA1 and BRCA2, you do see 100% coverage and 100% concordance with what has been previously known about these samples. So 60 out of 60 mutations in BRCA1 were called, and 21 out of 21 mutations for BRCA2 were called. A high degree of uniformity, so we know that more than 70% of the amplicons show more than 1,000 reads, which is very high, especially if you're interested in calling low frequency mutations. And as a matter of fact, the frequency mutation that can be called is as low as 5%. So even if the mutations that you're interested in are present at a 5% frequency, you can still use that workflow to call these types of mutations. So the last example that I want to go through is how you could use the BRCA1, BRCA2 gene designs for your biomarker studies, specifically for disease stratification in clinical trials. So again, this is an example of how you can use the BRCA1, BRCA2 gene designs for translational research. Now, just to give you some background, it is known that current prostate cancer treatments do not address the heterogeneity of the disease. So it is known that prostate cancer is a very heterogeneous cancer that has, different, sort of, you can call them different subtypes of cancer within that same family of prostate cancer. And that molecular stratification based on genetic aberrations can address the heterogeneity of prostate cancer. So instead of looking at prostate cancer as just one big disease, the idea here is to use molecular stratification to try to define those little subsets of prostate cancer and treat them differently. Now, what is needed are biomarker studies that can link the treatment response to these genetic compositions of the different forms of prostate cancer. And we know that BRCA mutational status has recently been linked to responses to PARP inhibition. This is exactly what I was talking about earlier about olaparib. It's known that the mutational status of BRCA can actually help us in these molecular stratifications. So we can look at the BRCA1 and 2 mutant ovarian cancer samples in a different way than those that have wild type. And we know that the BRCA genes are tumor suppressor genes and that are involved in the DNA repair machinery. So the question here that the authors wanted to do in this paper, they hypothesized that DNA repair defective prostate cancer is more sensitive to PARP inhibition than DNA repair intact prostate cancer. This paper was published just this past week in the New England Journal of Medicine. It's an excellent paper because it's a phase 2 clinical trial that actually stratified patients based on the genetic makeup of 113 genes. But so the idea here was to build on the earlier studies that I've showed for ovarian cancer, where the authors are asking this question, do we see the same happening in prostate cancer or not when these patients are treated with olaparib? And in order to do so, they developed a biomarker list that can be used to predict these responses to olaparib in a phase 2 clinical trial. They used a list of 113 genes commonly aberrant in prostate cancer, and these genes, they fall mainly within the DNA repair class of genes, including BRCA1 and BRCA2. So when they looked at the progression-free survival of the different patients, again they classified the different cases into biomarker positive prostate cancer and or biomarker negative prostate cancer. And when they looked at those cases that have been treated with olaparib, they were seeing a higher progression-free response rate compared to those cases that were treated with olaparib but that were biomarker negative. With a which was highly statistically significant. And when they looked at the different types of genes that have been shown to be mutated in these samples, if you look at the list of genes, you'll see that the majority of them, if not all of them, fall within that DNA repair machinery list of genes. So you see a lot of mutations in BRCA1 and BRCA2, in CHEK2, in ATM, in FA, NCA, in PALB2, and HDAC2, and so on and so forth. But basically, those that fall within this classification or this signature of these response genes, you see that these patients respond more favorably if they have mutations in those genes compared to those that do not have mutations in those genes. So to close this off with a summary, basically the BRCA1 and BRCA2 panel that QIAGEN has built delivers 100% coverage of all the bases. It delivers outstanding sequencing metrics, 99% specificity and 98% uniformity as defined by 0.2 x of the mean. It's a streamlined workflow with integrated data analysis and biological interpretation. So not only do you get to prepare your samples with a high degree of specificity and uniformity, but also you get to analyze the data with powerful tools and biological interpretation as well. The BRCA1 BRCA2 panel is ideal for the detection of both hereditary and somatic mutations. We know that both are important from a cancer perspective, and you can use the same panel for hereditary and somatic mutations, and you can use the same panel for different types of samples, including FFPE samples. The BRCA1 and BRCA2 gene designs, regardless of the panel, can be used for different applications in clinical and translational research. I've given you examples of how you can use it for routine testing, whereby you're screening or you're looking for specific mutations, known mutations in your samples. But you can also use the same gene design, whether it is in the BRCA1 BRCA2 panel or an extended panel, like the panel that contained 113 genes. You can use the same gene design for different applications in clinical as well as translational research. So I want to thank you again for attending this webinar, and I will turn it now back to Soon.
Thank you, Dr. Samara, for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. The first question is, Dr. Samara, are BRCA1 and BRCA2 genes involved in other cancers?
Yes, as a matter of fact, there was a recent paper that was published in the New England Journal of Medicine that showed how BRCA1 and BRCA2 and other DNA repair genes are also involved in prostate cancer as well.
Great. The second question someone asked is, can the panel be used for liquid biopsy?
The panel can be used for liquid biopsies because it generates small amplicons, so it is very compatible with the fragmented nature of liquid biopsy DNA.
Great. And what depth of coverage is recommended for this panel?
The depth of coverage is going to depend on the application. We usually recommend 200x if the user is interested in looking at germline mutations, and we recommend anywhere between 1,000 and 1,500x if it's for somatic mutation.
Can genes be added to the panel?
The panel by itself is a fixed panel. However, you can take the designs of the BRCA1 and BRCA2 panel and use them in a custom design where you can add as many genes as you want, up to maybe 160 genes.
Very nice. And we have time for one last question. It's nice presentation, Dr. Samara. The reference DB set looks fair. Wondering about the Ingenuity systems background. Is it based on MongoDB?
The Ingenuity system database is a manually curated database. It's been curated over the past 10 years or so. So it draws a lot of information. It draws its information from all the publications that exist in NCBI, but it also draws some information from a lot of databases that already exist, like the COSMIC database or the ClinVar database, and so on and so forth.
Great. If there are no more questions, I'd like to once again thank Dr. Samara for his presentation. Do you have any final comments?
Just as a final comment, the BRCA1, BRCA2 genes are becoming very popular when it comes to ovarian and as well now prostate cancer research. We know that they are heavily involved in responses to specific therapies like the PARP inhibitors. So in determining the mutational landscape of BRCA1, BRCA2 in specimens from ovarian cancer as well as prostate cancer will be very important.
Well, thank you so much for your presentation. Today's webcast will be available for on-demand viewing through 6 months from today's live date, November 11, 2015. You will receive an email from LabRoots alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.